Thank you, everyone. Today we have Dr. Eric Raabe from Johns Hopkins University. He's going to talk to us about his work on translational studies supporting the Pacific Neuro-Oncology Consortium adaptive clinical trials. Eric's talk is going to focus on the current clinical trial landscape for aggressive pediatric brain tumors. He will be highlighting the risks, opportunities, threats to the advancement of new agents to clinical trials. His talk will focus on highlighting the factors for the high failure rate of pediatric oncology clinical trials. And at the end of the talk, it'll help the audience understand the need for rigor, reproducibility in pediatric preclinical data before advancing drugs for testing in children. A brief bio on Eric. He received his MD and PhD from University of Cincinnati College of Medicine. He completed his residency at CHOP and worked in Africa as part of the Baylor International Pediatric AIDS Corps. He received his fellowship training following this at Johns Hopkins in pediatric hem-onc and pediatric neuro-oncology. And he has been a faculty at Johns Hopkins since 2011. Eric has been an ALSF researcher and has been funded through our continuum of research projects. He started his association with ALSF as a young investigator researcher in 2011 and has been awarded the Early Career A Award, the Accelerator Innovation Awards, and Reach Award subsequently. We are honored to have Eric here today. Eric is going to present his work to us, and at the end, you can type your question and answers. There is a little button. If you click on it, you can type your questions there, and Eric will address all the questions at the end of the talk. Thank you, Eric. The floor is yours.
Well, thank you so much, Vidya, and Liz and Jay and all the ALSF folks for inviting me to talk, but also for the support you've given me over the years. And so part of what, hopefully everyone can see my screen here, part of what we're going to talk about is stuff that I've done and things that my colleagues have done in the lab, but also sort of the overall state of pediatric brain tumor preclinical and clinical trial advancement, and sort of the challenges and things like that that we're facing and how we might potentially overcome it. So these are some objectives. We'll see if I actually can reach those objectives at the end. We have a history of great success in pediatric oncology, but we also have some pretty notable failures that we can kind of get into. And then, how does preclinical work actually lead to improvements in clinical trials? And is there sort of a correlation between how robust the preclinical data is, that is the things that we come up with in the lab, and how well a drug might work in humans? Of course, there are many caveats there in terms of mouse to human translation and things like that. But one possibility is the reasons why a clinical trial might not succeed or might have the opposite effect is that maybe we didn't do all of the preclinical studies that we needed to ahead of time. And so there's a new clinical trial consortium, relatively new, that has started, that is trying to do early clinical trials in pediatric brain tumors. It's an international consortium. Even though its name is the Pacific Neuro-Oncology Consortium, it's actually something that is spread all over multiple continents now, and provides a platform for potential new clinical trials. So I don't have any financial disclosures. I will be talking about drugs that are not yet FDA approved, which is, I think, somewhat of the point. And these are my opinions. They don't reflect the opinions of Johns Hopkins or the Pacific Neuro-Oncology Consortium. So I'm not speaking for anybody other than myself. And so one of the things that is important to recognize is that our current state is that, pediatric brain tumors are driving pediatric cancer death. And so this is sort of the incidence rate of brain tumors compared to leukemia, lymphoma. And this is from the Brain Tumor Registry of the United States in a publication that came out a few years ago. And what you can see is that brain tumors are actually pretty common. They're in red at the top. And so, they're more common potentially even than leukemia. And so it's important to keep that in mind. And this shows the annual mortality rate of various tumor types over years. And the very left is 1970 and 2015 is on the right. And you can see that the purple there is leukemia, and you can see that leukemia is a great success story. So from 1970 until now, a really dramatic drop in the mortality. And to the point now where brain tumors that have had a little bit of a drop has not kept pace with that drop for leukemia. And you can see that the current mortality for brain tumors exceeds that for leukemia. And you can also see that lymphoma has improved, and soft tissue sarcoma has also followed more of the brain tumor line rather than the leukemia line. So plenty of work to be done in brain tumors in specific in terms of improving outcomes. And this has all been published by this group that has been sort of studying and following this brain tumor registry in the United States to kind of understand how things are going overall in a population-based system. And so there are some really bad actors, in the brain tumor world, including glioblastoma, this diffuse midline glioma, where essentially nobody survives, HRT, where, as we'll see, the babies who are affected, primarily babies affected, 2/3 of them will die. Recurrent ependymoma, essentially nobody has long-term survival, and recurrent medulloblastoma with the current therapy, really only about 1 in 10 patients will survive. So really, while we may do better up front in things like ependymoma and medullo, if it comes back, there's not much that can be done that's curative. And so, there's something called a SWOT analysis that people do in business, where you talk about what your company's current strengths are, what your weaknesses are, opportunities, and then sort of external threats. And so we'll kind of think about that a little bit from our brain tumor clinical trial and preclinical landscape. And this is an MRI picture of a patient with a DIPG that's basically, or diffuse midline glioma, that's spread all throughout the pons, showing you why it's so hard to get rid of this tumor. It cannot be resected because it's basically infiltrated far and wide within the pons. And actually, if you were to go rostrally, you would see it would have spread even further, for example, into the midline structures of the brain. So one of the strengths that we have, and this is really one of the cool things about the last 20 years, really the cool things, is that we know more about the molecular drivers in pediatric cancer in general and pediatric brain tumors than we ever need. And that's great. And some of the earliest things were, for example, Jackie Beagle's discovery that the INI1 chromatin remodeling gene is mutated in ATRT. And this was published in 1999. And this was one of the things that actually really started some of the molecular revolution in terms of defining ATRT as a distinct molecular subtype of aggressive pediatric brain tumors. And that's been built on more and more. And so here's a paper that was 21 years later, but really describes how the ATRT, or atypical teratoid rhabdoid, is subtyped into different subgroups of ATRT based on methylation profiles and expression profiles of what mRNAs and what proteins they express. And this kind of just shows you that there are at least three specific groups of ATRT. And so then the question becomes, well, is that something that we can exploit therapeutically? And the same thing has happened in medulloblastoma. Perhaps, this is expression profiling. That was one of the first papers from Michael Taylor's group that showed that medulloblastoma exists in at least 4 subgroups. In high-grade glioma, Stefan Pfister's group at DFKZ showed that there are multiple subgroups of adult and pediatric high-grade gliomas, and that these are molecularly defined and are associated with outcome and location. And so they're at the top is sort of like the summary, if you will, of their work. And you can also find this, for example, in ependymoma as well. And then also in low-grade glioma, where there are key alterations in the gene called BRAF, which can drive the growth of low-grade glioma and could potentially be therapeutically targeted. And so, based on all this molecular profiling, we should be really poised for clinical progress. And so this was a review that came out quite some time ago, probably 10 or more years ago, regarding medulloblastoma, written by colleagues at St. Jude, which essentially said, hey, we have this molecular biology, but will it ever make a difference to clinical management? And that's really the challenge is well, how can we translate that we know a lot about your pediatric brain tumor, and we're going to know even more molecularly, but how do we turn that into clinical progress? And so there have been some really great success stories. So for example, in the low-grade glioma type, where patients have a BRAF V600E mutation, this is an activating mutation that also occurs in melanoma and other adult type cancers. And so pharma got quite interested in this and developed molecular therapeutics that would target this alteration, this BRAF alterations, including dabrafenib and trametinib. And so this is a clinical trial that was open here in the United States that showed some really great results. And so this is just one of my patients who was enrolled in this clinical trial. And before, she had this really big BRAF V600E mutant glioma in the midline that was causing her to essentially lose all sight. She was completely blind in one eye. She had we call field cuts in the other eye, and was actually having to hold her phone right next to her left eye to actually be able to see, text friends, which is for a teenager is really important. And she had to really enlarge the font size on her phone to be like headline font. For those who are young, maybe you don't know that you can do that. For those of us who are middle-aged, it's actually key to not have to put on our glasses all the time. But for someone like this patient, it was key to be able to communicate. And so she was at risk from this tumor, which was refractory to traditional chemotherapy, losing all sight completely and having additional complications. And so she was exactly the right patient for enrollment in this trial. And she started on the drug and had a few dose reductions due to toxicity, meaning like her had some skin rashes and other things. But this is what happened after just a few months on the drug. You can see that this turned into this. And when I saw her in clinic, she reported, oh yeah, I'm using my phone with regular type now, and I can read the menu at school just fine. And this was amazing. This was even before we had the MRI. So this is really remarkable. We got this really nice reduction, and even more remarkable that she regained a lot of vision. Now she's a college student and studying graphic design. So she's using her vision to actually create art, which is something that's really cool and something that would not have happened if we didn't have drugs that were primarily designed for melanoma, but which pharma decided to do a pediatric clinical trial focusing on this tumor type with the BRAF V600E mutation. And it's not just limited to low-grade gliomas. There's also been remarkable responses, as shown in this paper, to a different inhibitor called venurafenib used as monotherapy, where a recurrent BRAF V600E high-grade glioma was then treated with venurafenib, and the tumor essentially melted away over time. And that's really exciting. So that tells us if we just get smart enough, maybe we can find more targeted therapies or more therapies that will act like turning off a light switch that will improve outcomes in pediatric brain tumors. So now this is sort of answering that question that Drs. Gilbertson and Gajar asked, in that prior review, medulloblastoma, will molecularly targeted therapy ever matter? And so this is a Children's Oncology Group nationwide clinical trial where patients who have average risk, this Wnt subtype medulloblastoma, will actually have reduced therapy. And that's an important thing that needs to be assessed clinically. So the patients with Wnt actually subtypes did very, very well with higher intensity therapy. And so there was a question of, well, if you're able to cure 90% of the patients with the standard risk therapy, could you in fact reduce that intensity and still have that same cure rate? And so that is something that's being done in the clinical trial. And it's, of course, something that is fraught, because when you tell a parent, well, we can have a very, very high chance of curing your child, But we know that the therapy would be toxic and bad. And so what we want to do is try to reduce that therapy. But we don't know if that's going to be enough to cure a child. That's tough, especially tough because the prior children's oncology group, medulloblastoma standard risk clinical trial, attempted to do this across the board, not in a molecularly stratified way, but just across the board, and actually showed that if we reduce radiation in standard risk patients with medulloblastoma, there were more patients that relapsed and died in the reduced radiation group. So this was the case where essentially the take-home message for the non-WNT patients and most patients with standard risk medulla was that reducing radiation without coming up with something else to treat there was not going to be a successful strategy to maintain acceptable cure rate for those patients. And remember, when medulloblastoma recurs, generally the patients do not survive. So this is why being a clinical trialist is not for the faint of heart, because we don't know how this trial is going to turn out. And there may be patients who would have survived if they were treated with the standard risk radiation and a little bit more chemo who might not survive based on this trial. So we're going to have to see how this turns out. So it's great that we're doing more molecularly targeted clinical trials, and it's great that we have some agents that we can use that might target some of the alterations in pediatric brain tumors. But the brain tumor space is like super challenging for multiple reasons. And so one of the reasons is that unlike a sarcoma, for example, which often can be removed, resection can be really difficult. Now, our neurosurgeons are superstars and oftentimes can fully resect tumors, but tumors are in the brainstem, like that DIPG that I showed before. Low-grade gliomas, like that low-grade glioma I showed, that's also in the midline, this cannot be cut out. And so you can't really get that much out. And that means there's a lot of tumor that's left. The brain penetration of our drugs often is really poor. Our brain does a really good job of keeping things out of the brain so that we're not always being affected by what we eat. And so that then limits the penetration of drugs into the brain. And then one of the really interesting things is that it's very hard sometimes to get a response assessment because you can't actually look, you're just looking by a scan, and the response assessment can be really tricky because there can be times where the treatment seems to make the tumor grow, it's called pseudoprogression, and there's other times that treatments can make the tumor look like it's getting smaller, called pseudoregression. And there's a whole body of research and literature in neuroradiology to try to figure this out, to try to understand when does it actually mean that a patient is progressing, when does it actually mean that their tumor is getting smaller. So all of these things are, for pharmaceutical companies, reasons to stay away, right? So you may have a great drug that may work really well and hit a target really well, but there's a whole question of, does it get into the brain? And then another question of, how will we know whether this works or not? We have to wait for the patient to die, which would be, adding expense and time to any clinical trial, or is there really going to be a robust response assessment that you can do radiographically to give you sort of a sense of progression-free survival? Another problem in pediatrics, which is something that can be difficult to address, is that thankfully our tumors are relatively rare compared to adult patients, right? So that is great that there's not that many kids who get ATRT every year. And so that means that to enroll a clinical trial of ATRT patients, you really need a multi-center undertaking, and the trials have to take a long time. And so that is an example of this, where it was successfully undertaken, is the last CRG HRT clinical trial, which the concept was started in 2003. The ACNS 03 tells you like it was started in 2003. And it's actually started accruing patients in 2008. So it took 5 years to get through all the approvals and everything and to get the green light to actually launch. And then the data was published in 2020. So this gives you an idea of how long it will take for us to actually learn from some of our clinical trials. In this case, from 2003, where it was an idea and a concept, to 2020, so 17 years. And this enrolled 65 patients. I think 70 were enrolled and 65 were actually filed for outcome. Some were ruled ineligible. So, but this is the largest and most robust HRT clinical trial to date, I think. And the results are quite good. If you look at the historical control, you can see that before sort of the advent of high dose chemotherapy followed by focal radiation, which most of the patients on this cohort received, the outcomes were abysmal. In fact, so bad that in some cases, patients were told that they shouldn't pursue, or families told they shouldn't pursue treatment for HRT because it was essentially hopeless. Now, with this 0333 backbone, we can say, hey, there's a chance that a child will survive and be cured essentially with this. And here you can see most of the relapses happened in the time since enrollment. Essentially by 2 years, there are very, very few relapses that happen after that. And all these tick marks are patients who are lost to follow-up. So, and this shows you the difference between overall survival and event-free survival. So this is really a great success and really a testimony to Alyssa Reddy and all of the people who worked on this trial, their staying power in terms of being able to design it and then stick with it and analyze it at the end. And Alyssa was when I was going to COG, she would show sort of pictures of the clinical trial development, also pictures of her child as her child was growing up, as the clinical trial was growing up. I think it may have even had an in utero ultrasound to begin with. Well, here's my son when this trial was started, and then here he is 17 years later when this data is being reported. So that is an example of how the COG and the overall pediatric oncology group kind of overcame issues of small numbers, but 65 is still relatively small, and it actually is somewhat limiting still for the molecular analysis, the subtyping, to actually be robust enough to say, is there any specific subtype that does better or does worse in HRT? So still to be determined. And one of the other weaknesses that we have, to change gears a little bit, is that we'll do a clinical trial through CRG or other places that will have pretty good data, and then oftentimes we'll decide not to use that data, which is kind of interesting. So you can see this in relapsed medulloblastoma, where Avastin, temozolomide, irinotecan actually did fairly well for patients with relapsed medulloblastoma, but that's not being considered further. And then for low-grade glioma, here's an example of carboplatin vincristine compared to thiaguanine procarbazine, lomustine or CCNU and vincristine, and the TPCV, even though the biostatisticians would say the Kaplan-Meier or log rank test is invalid because the curves cross here, additional statistical tests and just eyeballing it would say, well, you know, if you're on regimen B, which is TPCV, half the patients relapsed at 4.5 years, while if you're on carboplatin vincristine, it was like 2.2 years. And so that difference would suggest that regimen B might be superior, but regimen A has been advanced for future use in the children's oncology group. And part of that may be the difficulties in administering a mostly oral regimen to patients as opposed to an IV regimen. So there may be reasons, but you know, in this case, as one of our biostatisticians said, I know which curve I would want my child to be on. If my child were to have a low-grade glioma, I'll be regimen B. Similarly, in high-grade gliomas, there's a result of the ACNS 0423 study, which is a big study that combined lomustine together with temozolomide and compared it to historical control of temozolomide alone and showed that it was superior. And a statistically significant comparison where, you know, this is, you know, the combination of CCNU or lomustine plus temozolomide, and this is temozolomide alone, and you can see that you know, here it's not that remarkable, but it gets to be more remarkable as time goes on, that you have a good number of patients who have not relapsed at 2 years in the combination therapy, while the monotherapy arm is here. And of course, all these patients got radiation and resection up front. And it got even more remarkable, before I go on to the next thing, that patients had a gross total resection. This was all patients, but patients had a gross total resection surgically. Actually, they had quite a number of long-term survivors. And this is It's pretty remarkable that, with this regimen, you could see that 20%, 20% of the patients were alive at 3.5 to 4 years, which is not usually what we think of for patients with high-grade gliomas. So another weakness that we face is that, similar to what we figured out from molecular profiling, we have potentially new targets, but we also have a lack of new drugs. So this is something that our group found where we identified this protein Lin28, which is a stem cell factor, and it's expressed almost universally in ATRT or atypical teratoid tumors. And so there's not like a great group of drugs that are advancing to the clinic targeting Lin28. And so here you have a target, and one of the things that we've been trying to do in our lab is to come up with ways of targeting Lin28. So you have a pathway that you want to disrupt, but how can we actually drug Lin28? And so there are several labs that are working on this, but those things are far, far, far away from being in patients at this point. And so are there ways that we can look at the downstream effectors, such as the MAP kinase pathway or the mTOR pathway? And so that is where we kind of have put much of our energy with regards to ATRT. And this work was largely spearheaded by Jeff Rubens, who started out as a fellow in our group and now is faculty here at Hopkins. He was kind enough to share some slides with me. So when I say we, I mean Jeff, okay? So just keep that in mind. And so a good number of our group, Jeff and others, focused a lot of time on this drug called TAC228. And TAC228 is an mTOR inhibitor that hits both parts of mTOR. So there's TORC1 and TORC2, and TAC228 gets into the brain really well. And it's oral, and has a short half-life. So we thought, well, this could be a really good drug for us to try to use. And so we showed activity in low-grade glioma and in medulloblastoma and in DIPG and in HRT. So we thought this is going to be great. And so Jeff worked really hard in coming up with a clinical trial, working with the clinical trialists to come up with a schema that was supposed to be advanced through the Pediatric Brain Tumor Consortium, or PBTC, which is a NCI, National Cancer Institute funded consortium. And TAC228 was available from the company through the NCI, and they were going to do this sort of phase 1 A1B together with carboplatin for a lot of different patients with low-grade glioma, medullo, DIPG, and high-grade gliomas that were recurrent or refractory. Try to figure out the dosing and those kind of things. And this was like a lot of work, right? When I say this happened, so Jeff spent a lot of time on this, and PBGC really liked it, and the company seemed reasonably enthusiastic about it. But then the NCI decided not to move forward with it. And it's the NCI's right not to do that. They would pay, more than $1 million probably to run this clinical trial through this multi-site consortium. And so they had a right to do that. They said, well, you're not actually targeting the driver mutations, was one of the things they said. And as far as I know, that was the first time that the NCI had stopped a PBTC clinical trial that had been fully accepted and approved by the pediatric brain tumor experts at the PBTC. And then, of course, the company decided, unfortunately, not to pursue the drug clinically because it didn't have great success in the adult world, where the population was too small in the adult world. Then it was sold to another drug company called Calithera, and the future plans for that drug are unclear. So this kind of shows you the weaknesses, right? So we did everything we could to try to get this into clinical trials for patients, funded in part by Alex Lemonade Sand. We had a clinical trial that people thought was the right thing, and things fell apart relatively quickly thereafter. And so maybe it was the right thing for NCI not to pay $1 million worth of taxpayer dollars to fund a clinical trial for a drug that was kind of wobbly anyway in the adult world. But that then led us to kind of retrench and start a new thing. And so, we'll get to that in a second, but this gives you a segue to sort of what the opportunities might be for sort of moving things forward. And so one of the things that we tried to do with TAC228 was provide, robust preclinical data to convince the drug company to pursue the clinical trial. And we think we had pretty good data, but it wasn't enough, and there were perhaps other reasons that were out of our control. But that doesn't mean we should give up on this approach. And the real question that we always have to ask ourselves is, and this is a really important question for brain tumors, is the target, that is, we think we're hitting a target. We want to stop mTOR, we're going to shut it off, like turning off a light switch. Is that target a valid target in the brain tumor world? And is the drug able to hit the target? So administering a drug by mouth to a patient and then asking, does it actually get into the brain in measurable levels? And then does it suppress that target? We can do this in mice, but we're not really interested in curing mouse cancer. And I think it's really important to do this in mice. And Jeff had actually done this in collaboration with Kathy Warren when she was at NCI, with actually were able to measure levels of toxicity in the mouse brain and in the brain tumor and show that over time they were able to inhibit the mTOR pathway. So that was really important to do that in mice. But is there a platform to do this in human patients? And this gets to that whole question of providing benefit in a clinical trial setting to a child, but also learning something, right? So we have 30 plus years probably of clinical trials in diffuse midline glioma or DIPG, where Arzu Thomas, a biostatistician at St. Jude, showed the amalgamated Kaplan-Meier survival curves for this, and they all overlap. So 30 years of clinical trials, every patient seems to follow exactly the same path. That, what I mean is each cohort seems to follow exactly the same path, so that everyone essentially is dead by 2.5 years. And that is terrible, right? Because that means that all these clinical trials have done and nothing's really ever worked. And so, but part of the problem is we haven't had the ability to figure out whether the drug actually gets into the ponds, whether it actually hits the target. And so Sabina Mueller, who's one of the founders of the Pacific Neuro-Oncology Consortium, wanted to address this. And I've been honored to be sort of a small player in some of the diffuse midline glioma adaptive clinical trial group. And so watching this clinical trial get put together has been really great. And so one of the features of the clinical trial is multiple arms that can come in and out. So a new agent could be advanced with robust preclinical data. Patients could be given that agent, and then part of what they would like to do is actually to have the patient take the drug first before there's a biopsy, and then multiple biopsies are taken from the tumor and the pons, and some of those are used to assess drug levels, and some of them are used to assess whether the drug is actually hitting the target, suppressing whatever target I think. Because if the drug is not detectable in the pons, or if it is detectable but the target is not suppressed, then the chances of this drug actually working in this tumor type is very, very low. And so this is the real important question. So you might say, well, the target, whatever it is, mTOR, was suppressed and the patients all still relapsed or progressed the same. So maybe mTOR is not a good target to go for. Or you could say the target was suppressed or target was not suppressed, right? So let's just say the drug was given and there was no drug that was detected. So that means the drug is bad or not the right drug to be given to patients with brain tumors because its ability to get into the brain is poor. So if you give a drug and you can't detect the drug in the tissue, tumor tissue, and your target is not suppressed, chances are your drug is not getting where it needs to go. And so there are a series of these type of trials that are being worked on. And this is one of the first in the pediatric brain tumor space to actually be bold enough to propose to do this. And uptake in this trial has been actually really, really good. And DMG, as I showed before, or diffuse midline glioma by the MRI, is actually one where it's previously just been a diagnosis that you could have by the MRI, and then you go off to radiation and maybe get some additional chemo, but you didn't actually have to have a biopsy because it's characteristic MRI is so obvious. But because there are different subtypes of DMG and different potential targets, there's a ethical reason to do a biopsy, which is you would like to know, first, to confirm the diagnosis, secondly, to figure out by molecular profiling if there's any weaknesses or Achilles heels that you could target. So that then allows us, broadly speaking, the pediatric brain tumor world to take one or two extra cores to try to figure out if the drug is actually getting in and if it's hitting the target. So this is really a model that in the ATRT space, we probably will not be doing biopsies in the recurrent setting, but this adaptive clinical trial where the drug could advance, it's hitting the target, but the patients don't have long-term benefit from it, then it may fall out and a new drug could advance in without stopping the clinical trial. So you could potentially have multiple arms as well that could be open at any given time that would have certain molecular biomarkers. You have a particular mutation that then would cause you to get a certain drug. You don't have that mutation, you'll get a different drug. And so this is really a way of having a clinical trial be open to have us maximize how much we can learn about the tumor and about the drugs that we're testing, and potentially benefit patients. So could an adaptive clinical trial be useful in HRT? So this is what I mentioned, having multiple arms that would allow us to test drugs with robust preclinical activity in children, and then identify active agents for potential further exploration. And then what's also great for patients is that they could, if we had multiple arms open, you could potentially have a patient start out in one arm, arm A, for example, and then they got benefit from it for 6 months, or then had some toxicity and had to come off. They could potentially transfer to arm B and still be in the same study. So that part is pretty exciting. And this PNOC is an international clinical trial consortium, and so children all over the world could enroll. So it's open in Australia, Israel, Egypt now, as well as, of course, North America and Europe. So lots of opportunities to find patients who have HRT and help them, and to help us learn more about this disease. And the folks who are really spearheading the clinical side of things are Ashley Margol at the CHLA and Alyssa Reddy, who's at UCSF. And so these are the folks who are really developing this adaptive clinical trial protocol. So I very much appreciate my clinical trial colleagues who are helping with all this. And so to talk just a little bit about the robust preclinical data that we have developed that would, we hope, allow us to activate the first arm of the study. This is, as I say, Dr. Rubin's data. He's been kind enough to share it with me, and we've been combining this drug called Paxalosib with a traditional chemotherapy called gemcitabine. And as we kind of had already alluded to, there is some pretty significant papers existing already from our group showing that TAC228 as an mTOR inhibitor was active in HRT, and Paxalosib is a PI3 kinase mTOR inhibitor that also has great brain penetration, and is currently in clinical trials for high-grade glioma and in DMG. So the company believes that this drug has really good brain penetration and would be appropriate for testing in HRT. And interestingly, as part of our collaborations, globally through the miracle of Zoom, we're able to have these meetings where the Europeans at 11 o'clock at night are able to join. And some of Esther Hilleman's group had pointed out to us that they found that gemcitabine was actually a very active agent in HRT. And so that they suggested combining Paxalosib with gemcitabine. And so Paxalosib by itself has some activity as a single agent in multiple models. So this is the Paxalosib arm here in an aggressive HRT model called CHLA-6, BT12. So there's definitely a survival advantage. This is the bioluminescence imaging showing that the controls tended to progress while the majority of the Paxalosib treated mice had stable disease. There's always one that's an outlier, but that's perfectly fine from a biological heterogeneity standpoint. And then combining Paxalosib with gemcitabine seemed to activate this integrated stress response and lead to apoptosis. So CHOP, ATF4 are the readouts of this integrated stress response. EIF2 alpha is another one of the readouts. And so when you add Paxalosib and gemcitabine together, you got a lot more apoptosis, and this is in vitro. So that was really exciting. And you can see, you don't have to be a scientist to see that there's a lot of cells in the control here and not that many cells in the combination. This is a fancy measurement of synergy, which is a mountain plot showing that the higher you're in this direction, the more synergy you have. And synergy means 1 + 1 = 3, essentially. So a little bit of apoptosis in this sort of realm and a little bit of apoptosis there leads to a lot of apoptosis in the combination. And these are very high synergistic scores, suggesting that the two drugs would work together very well. This is another way of looking at it in a different cell line where we're combining Paxalosib and gemcitabine, showing a lot of apoptosis in the combination compared to the control or the single treated. And then a very good synergy score again of Paxalosib and gemcitabine. Different cell line showing essentially the same thing. And then once again, a third cell line, so this is this idea of having rigor and reproducibility. And this cell line has potentially less mTOR activation than some of the other ones, But still, you can see there's a good synergy score at the peak synergy area over here. That's acceptable, and a good induction of apoptosis. So this could be something that would be active across the subgroups of ATRT. This BT37 is thought to be a different subgroup from CHLA-6. Again, this is all in vitro data. But the real importance is the in vivo data, where you can see that if you're a mouse, you really want it to be in the combination group, because we had to reduce the dose of Pax-ALSF a little bit to make it tolerable together with gemcitabine. So Pax worked a little bit less well than it did before. Gemcitabine by itself worked very well, but the combination is continuing to do very well. And you can see that we're basically at the 50% and have essentially quadrupled survival, which is a good indicator, we think, that this is a highly active agent. And then a similar result has been shown so far in BT37, which represents a different subgroup, as I mentioned, from CHLA-6. So again, a really nice, single agent effect of Pax-ALSF, single agent effect of gemcitabine, but the combination is really going strong and looking very good now, 100 days. So we're hoping that this will similarly lead to significant extension of survival. This is the bioluminescence imaging after 5 weeks, showing the controls will have these really robust tumors. The combination has relatively small tumors. So really exciting potential data. And when you see activity of a combination in multiple cell lines and then in vivo in multiple orthotopic xenografts in the brain, this is really telling us that we think it's getting into the brain. We think it's actually hitting the target in the brain, and we'll have that data to show as well, at least in the mouse brain, that we're able to do that. So we're going to keep going with more orthotopic models to convince clinicians that this is something, and parents, that this is something that they should try for patients with relapsed ATRT. And then, we want to really try to figure out what are the biomarkers that would predict sensitivity. Is there a particular molecular marker that we could look at in their initial resection that would say, oh, this is the right drug combination to use for these patients? And then what's going to happen? What's happening in the mice when they actually do succumb, if they do succumb to this combination? Will we cure mice, or will there actually be some that will come back with resistant tumors? So those are some of the opportunities, and just to highlight those things. And so now to get to the end of this, the threats. So, when you think about it, there's not much motivation for drug companies to undertake clinical trials in pediatric patients. I mean, of course, they care about kids, but what they care about mostly is not having an adverse event that affects the approval process for their drug. So if your drug is not FDA approved yet, the last thing you want to do is expose a child to it and have the child die. Of course, because that's terrible that the child dies, but really what that does is gets the FDA really worked up. And then that potentially puts a hold on the clinical development of your drug. And it doesn't have to be death. It can be a bad outcome, whether that's a new tumor, whether it's a terrible side effect. And so what's the plus side, right? So what's the plus side for them to actually do clinical trials in patients who are children as opposed to adults? Thankfully, the numbers are very small for pediatric cancer, but that also creates a big financial problem. And so we'll talk about that in a second. And doing combination therapies with novel agents can be really difficult, because if two companies are both working on trying to get their drug approved, and then there's some side effect, everyone's pointing at everybody else, but the FDA is pointing at both of them. And so if one company owns both drugs, that's great. But you have to think, if we're actually going to make an improvement in outcomes, it's very, 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 very rare, I would say, that any pediatric cancer is really cured with single agent therapy. Now, we've seen a couple of cases that are celebrity cases where venurafenib as a single agent caused that tumor to shrink down. And you'll see that there's, for certain fusions, that's like a light switch, turn it off. But is that durable, and are the patients cured? Because certainly there are almost no leukemias and no sarcomas that are cured with single agent therapy. The only thing I can think of is CML, where, there's one drug, dasatinib or imatinib, that turns off the abnormal receptor tyrosine kinase, and then that leads to regression of the tumor. But for the most part, to actually cure a patient, you need multimodal therapy. We talked about the financial incentives a little bit, and so Nancy Goodman at Kids v Cancer has been working very hard to try to come up with strategies to incentivize drug companies without harming the taxpayer to to do clinical trials in children. The FDA has a requirement that drug companies have to at least have a plan to develop their drug in children if it could potentially be a helpful. So that's something that is probably very helpful. The drug companies are looking to do phase 1 clinical trials. However, once they do a phase 1, their interest in continuing that may be modest at best, for all the reasons that they might need to develop a syrup because kids can't take pills, for example, or you need a different formulation for children for that reason, or an IV vial might not be the right size. Children oftentimes have increased metabolism, so phase 1 studies sometimes show that the drug needs to be a higher concentration than adults. Targets can be really difficult to disrupt, so fusions or epigenetic drivers as opposed to point mutations. And even in adult high-grade glioma, talking to my adult GBM colleagues, they don't feel that their pipeline of new drugs to try is particularly robust, and many pharma companies just stay away completely because it's so hard. It's so hard to get a survival advantage in adult high-grade glioma. And lastly, there's really uncertain funding for clinical trials and particularly preclinical studies in pediatric cancer, right? So like everyone wants to come up with a new mechanistic whatever that will set the field on its head. And this is going to be this really great thing, but the translation time from a new mechanistic study into a patient is 10 to 15 years probably, and maybe 20. And to actually get into children, probably 20. And so that's kind of like, funding things that are, wow, we're really going to unlock this at the expense of, let's take a drug that already exists and prove robustly that it works. It is kind of like wishing upon a star, as opposed to taking an existing drug and figuring out how to make it better together with traditional chemotherapy. So that's an example. So I know we're running low on time, so I will, instead of talking about threats at the end completely, I will just talk about how we've made so much progress. And here's some cool matchmaking between a molecular study or several molecular studies, one done at JFKZ at the bottom, and that basically found that there are these novel fusions in babies with high-grade gliomas, and that these babies that have, for all the world, what looks like GBM actually can have a very favorable outcome. And so understanding this, that a glioblastoma in an infant is not the same as a glioblastoma in a 4-year-old or a 14-year-old, and certainly not the same as a 40-year-old, has led to some real revolutions in the way that we can treat these patients. So they can have these huge tumors, right, at diagnosis that then could be resected as best as possible by the neurosurgeon, and then get traditional chemotherapy and not radiation. And then you can see that it shrinks down quite a bit, and then they stop chemo and it starts to grow again. They have another surgery. They start some more chemotherapy, and now the chemo is not working. But then they start this targeted agent that disrupts this ALK fusion. It turns out that ALK is frequently altered in neuroblastoma in children, but also in some types of lung cancer. So drug companies have made drugs that target ALK fusions, and they made drugs like ceritinib that get into the brain because some of these patients with lung cancer have brain metastases. So now here's the matchmaking between a drug, ceritinib, which is FDA approved, and a patient who has a potentially novel ALK fusion, where the patient has durable benefit from this. And so that is sort of what we're hoping for. And if you could move this together with the traditional chemotherapy up front, then you might very well have a situation where you might actually not have the patient recur, and you might very well be able to cure them. And finding out these sort of things is what we spend all of our time doing, taking the molecular information, finding matchmaking with drugs, or developing new drugs to then get into the patients to have a better outcome. So with that, I'll thank all the people who did all the work, as well as our funders who are here. Particularly, I want to thank Jeff Rubens, who really has been leading the way in the HRT world, and then Charles Eberhardt for being a great neuropathology collaborator and research mentor, and then everyone at the PNOC HRT working group, and most importantly, Ashley Margol, who's taking the lead there. So with that, I will stop the share, and I will turn on my video so you can see once again what I look like, and then we can potentially see if there's any questions.
Thank you, Eric. Great talk. Thank you so much. We do have a few questions and I can read them out to you. The first one is from Liz Scott. Would placement into the arms on the adaptive trial be based on each child's characteristics, or is it random?
Yeah, so that's a great question, and some of it will be the answer is it depends. So what we would love to be in a situation where we would have an abundance of arms to offer patients at the same time. Just based on all the factors that I mentioned right there, chances are we're going to open it with one arm to begin with. And our lab is currently working on arm 2. And we're also open to hearing from other investigators. So it's not just like I'm not the only one, but so if there's an immunotherapy arm, for example, that looks really promising for ATRT, people bring that to us. We would love to be able to incorporate that, and they don't have to be part of PNOC. We're excited to, our meeting is open actually to all investigators. So we have folks from Denver Children's who are part of it, and they're not part of PNOC. And so, we're really open to having more input and more arms. And so, ideally we would have multiple arms, and we would be able to sort of do matchmaking, as I say, and say like, okay, well, you have your tumor, when we originally resected it, has this whatever marker that will predict response to this immunotherapy. So we're going to prioritize that one for you over sort of the ALSF gemcitabine arm. And we'll see how that goes. Versus somebody else who maybe doesn't have that marker, but has really high activation of mTOR in their tumor. And say, well, that you would be perfect for this ALSF arm. So that is still to be worked out. Right now, it's essentially, we're going to have one arm to open, another arm hopefully to follow shortly thereafter, and then additional arms will come in and out as time goes on. Some will be molecularly stratified.
Okay, thank you. Next question is from Robert. Hi, Eric. Great talk. Thank you. What are your thoughts about duration of use of some targeted agents? We don't know when to stop some agents like dabrafenib. In case of TRK inhibitors, trying to stop or change inhibitors can lead to rapid progression.
Yeah, and this is really, there's a lot of ways that you can have a wrong answer here. And so bringing up the TRK inhibitors is really important, because there's a big push from a lot of people to say, well, maybe we don't need to treat for quite as long with MEK, dabrafenib and MEK inhibitor. For gliomas, and maybe we can do 9 months instead of more than that. And so in the patients that I've treated with dabrafenib trametinib, their vision has been recovered, so in a couple of cases, and that's been great. And so one patient had to stop because he had hyperglycemia, and so it just so happened that he came off the drug. We got him his blood sugar controlled, and he was due for an MRI on the study. And of course, the enhancement got bigger, but so did the overall tumor. And so whether that was because the blood supply changed, and that led to more microcysts blowing up, or who knows what. I'm kind of waving my hands because we really don't know. But in that particular patient, it kind of had me think, well, he's gotten a lot of benefit from the drug, and we just have a little bit of evidence already that his tumor is not going to stay quiet if we take the drug away. He has a BRAF V600E with a P16 deletion, so exactly his worry would be bad. And so his side effects are minimal, and he's doing very well. So from that standpoint, I'm inclined to keep him on. So he's been on for several years. For patients who are having bad side effects, oftentimes it makes total sense to stop 9 months in. For low-grade gliomas, we're not seeing a lot of what would be considered true resistance, I think. For high grades, there's a good body of literature coming from Denver as well as Hopkins that autophagy is increased with dabrafenib trametinib, but that's sort of an escape mechanism. And so one could add things like chloroquine to dabrafenib trametinib and sort of restore that sensitivity. But again, this is the $1 million question of when can you stop some of these targeted therapies, if ever.
We have time for one more question. Thanks for a great talk, as always, from Shubin. What is the next best step once we have a drug that we demonstrate is efficacious in preclinical models?
Yeah, so it really, I mean, it's matchmaking as well, because you have to have a drug company that's willing to at least give you the drug, right? So that in the type 2 weight setting is a great example of, we had done quite a lot of work. We, meaning Jeff, had done a lot of work on this, and we thought we had pretty good, preclinical data, but the drug company was very wobbly with the drug in general, and in the end, that drug's probably not ever going to make it into pediatric patients, which is unfortunate. But thankfully we have ALSF. So I think having a robust target that you believe is a good target is really helpful. And so then, if not, drug A, then hopefully drug B will come along, and publishing your papers will hopefully be a signal to a drug company that maybe there's some interest there. And in terms of Chiba and your work, right, and things that we're working on in terms of the MAP kinase pathway, we may finally have a MAP kinase targeting drug that might actually do something in the brain. So we're well on our way to hopefully following up on all the good work that you did, eventually to be able to really translate that into patients.
Okay,
Whether
thank you.
the company is going to want to do the trial is a whole other question, though, right? And that's always the thing, is that we just don't know whether they're going to say, we've done our one pediatric clinical trial, and then we're not interested in having the risk or coming up with a new formulation for kids, or whatever that might be.
Thank you so much, Eric. This was great. We really enjoyed your talk, and thank you for being with us today.
Well, thank you for having me, and thanks everybody for joining in. And we're always eager to hear, and have more conversations about this kind of stuff, because there's the only way that we're going to improve our outcomes from that very flat curve is to do more research. So thank you all. Thanks to Alex Lemonade Stand for supporting all this work too.